So good morning and good evening, everyone. This is Wang from the Institute of Molecular Biology, Academia Sinica. I'm the host of today's seminar, and we are happy to have Dr. Andrew Wang at Yale University to share his recent findings. Dr. Wang received his bachelor's degree from Harvard University with a major in biochemistry. He then moved to UT Southwestern, where he conducted his thesis research and received the MD PhD degree in 2011, with additional training at the University of Paris. During his MD PhD training in the labs of Dr. Edward Wigland and Chandra Mohan, Dr. Wang discovered the crucial role of the CXCR4 SDF1 axis in end organ targeting in SLE. After finishing the internal medicine internship and the residency at Yale, he joined the ABIM short track pathway into the rheumatology fellowship. In July 2014, Dr. Wang began his postdoctoral training with Dr. Ruslan Majitov, where his focus shifted to understanding the coordination between inflammation and metabolism. This research led to the discovery that various inflammatory states are associated with different metabolic programs, a crucial insight into the pathogenesis of various inflammatory diseases. Dr. Wang joined the faculty as assistant professor in internal medicine rheumatology in August 2017 and the immunobiology faculty in July 2019. His lab is focused on exploring how the environment impacts disease progression by incorporating patient samples and using a range of techniques from disciplines such as physiology, metabolism, inflammation, neurobiology, and immunology. So let's welcome Dr. Wang for his talk.
Hi, Wayla. Thanks for that kind introduction. And thanks to Dr. Wu for inviting me to speak here. Okay, can everybody see the pointer? Wayla, can you see the pointer?
Yes,
okay.
I can see I can point.
So here are my disclosures. I have no conflicts for this particular presentation. As Wayla mentioned, part of my lab is interested in trying to understand the mechanisms that drive phenotypic heterogeneity. And so today I was going to present some of the work that is mostly unpublished. And please stop me at any point if something's unclear or if you want to ask a question. So I thought I would start with an example that's very famous during the time of COVID. And I think it highlights the problem that we're interested pretty well. So this was a case report out of the Annals of Internal Medicine. It described an Italian twin pair. So they confirmed this genetically. And they lived in the same place. They worked in the same place. They got COVID from the same person in their workplace, showed up in the emergency room on the same day, that is same cycle numbers by PCR. And then one of them walked out and the other one had very severe disease in the ICU. And so if you look at their table 1, where they have all of the patient characteristics and all the things that one could easily observe, they were for the most part the same. And having served multiple tours in the acute medicine service in the COVID wards, this is something that we saw a lot, where 2 very similar patients in a lot of their characteristics had vastly and dramatically different outcomes. In genetics, this is called incomplete penetrance. And so here are a group of diseases that I see in my clinic in the ambulatory setting, autoimmune diseases, where what people have found is that the concordance rate, so where both twins have these diseases, lupus, rheumatoid arthritis, is significantly higher by a log fold than dizygotic twins. But you can see that the penetrance is incomplete. This is true for neonatal sepsis. It's also true for allergic diseases. And in general, patients will then ask you, well, what, especially in counseling for having children, what's the risk of me passing this on to my children? And I usually refer to them to this concordance rate data. And then they'll ask me, well, what's the rest of the risk? And then you say the environment, And then they generally ask you, what in the environment matters? Should I move to California or Florida or whatever, and be vegan? And the fact is that we don't know. In the laboratory setting, it becomes a little bit harder to understand lethal dose 50, where now you have genetically identical mice. They're in the same cage, they're on the same rack, and you can give them the dose of something. So in the top case is LPS, and the bottom case is anaphylactic shock. And you can kill half the mice. And it's really not intuitive how this works. This was actually described in toxicology all the way about a century ago by Yale. And you can see that in the end, when he's talking about using laboratory animals to determine lethal dose 50 so that we can find safe levels of drugs to give people, he had a lot of consternation, a lot of anxiety about how this works, and talks about a lot of possible items in the discussion because it's not intuitive why this happens. And so the even simpler version of this is a thought experiment, very famous thought experiment by the physicist Schrodinger, when he was trying to think about kind of the stability of quantum states. And basically, the idea here is that the cat, it can be both dead and alive, depending on the state of a particular poison that that has yet to be released. So it's these kind of bistable states that I think are very interesting. And so our first approach to trying to understand the LD50 phenomenon was kind of a hypothesis-driven screen. And so what we did was we reasoned that even though the animals are all the same in the cage, and the cage environment is the same, not all the mice, for example, would have eaten their last meal at the same time. Since we house them, generally speaking, all males or all females for husbandry. For example, those of you who work with mice know that in the cages, there's always kind of an alpha male, and they'll beat up on a smaller mouse. So the level of kind of stress amongst them is different. And they don't, all huddle in the same kind of groups. And so basically, we thought of all the things that could be different and normalized them, and then asked if we applied this normalization, would we end up with all dead mice or all alive mice? And then did phenotyping and try to understand the biological pathways, and then to try to explain what was driving this vastly different outcome in a setting where the genetics and the environment are very, very well controlled. And so this work was done by my first postdoc, Dr. Ching, who's now a lead scientist at Genentech, and a very, very talented postbac who's now a PhD student at Yale, Reina. And basically what they found, they tried a lot of things. So they normalized for the last meal, for the thermal stress, for the psychological stress, et cetera. And basically they found that if they stressed out all of the animals equally before, they could kill all of them. And this was what ended up leading to the second paper out of my lab, where we found a very unexpected role for an inflammatory response secondary to psychological stress. The second approach was actually thinking about what in the environment could have changed. And the reason we looked at this is because of this graph here from Platts-Mills, describing an exponential increase in certain types of diseases as a function of time. So this kind of increase in disease prevalence for allergic diseases, turns out it's also true for autoimmune diseases. And so we think of these diseases as diseases of modern industrial environments. And so what a postdoc in my lab who's just actually joined the faculty, Anna, and a very talented postdoc, Brandon, who's an MD PhD student now at University of Colorado. What they noticed when they were thinking about what in the environment could matter was that a lot of industrial xenobiotics, so things like pharmaceutical medications, in the example of aspirin and nonsteroidal drugs that you can buy over the counter, as well as food preservatives for industrial food processing, were also added simultaneously. And so they asked a very simple question, actually. So unlike the hygiene hypothesis, in terms of thinking about why there's more allergic diseases now, they asked kind of the opposite question of, is it possible that we put into the environment something that now has adjuvant property that is making antigens that we did not have an immune response to prior to industrialization, now something that was antigenic, leading to allergy. And basically, I'll show you in the next slide what they discovered and how we ended up with this publication last year. But essentially what they found was that amongst these common drugs that we all use, many of which are over the counter, the NSAIDs actually had a very surprising property of activating a xenobiotic sensor, and different NSAIDs had the capability of activating this xenobiotic sensor differentially. And this work actually came out of them essentially doing what I showed you in terms of screening modern xenobiotics, which was if all they did was take ova and then use a standard adjuvant like alum, where they can sensitize mice to ova and make them make ova-specific IgE. Could they do the same thing if they just put in the industrial xenobiotics? So in this case, ibuprofen, Tylenol, they tested all kinds of NSAIDs. And then after they did this, they found that some of the NSAIDs could do this and lead to anaphylaxis in various routes, and some of them couldn't. And in the molecular dissection of this, this is how they discovered that the property of the NSAID was NRF2. So today I'm going to talk about kind of the results of our third approach. And I promise this is the last time I'm going to show such a kind of a complicated, busy slide. But this approach was essentially to still use the LD50 model, but then kind of try to reverse engineer it. And so the idea here was, can we find characteristics of mice that differed, that could predict if they would live or die. Of course, these type of things would have to be done non-terminally, so things that we could measure without sacrificing the animal. And then once we found them, validate their predictive power, figure out what the biology was that was driving whatever parameter it is that we found, and then kind of work backwards here and see how far back we can go in terms of when mice kind of diverged in their disease courses. And so the work I'm going to spend the rest of the time talking about today was done by my first PhD student who just graduated, and another very talented postbac, Michael, who's now an MD PhD student at Columbia. And would like to hear what you think about this work. So the basic biological question here is, can be summarized here, which is, are we starting out with mice that are all equally susceptible, and then you hit them with some challenge, and then because of random factors that occur after challenge, they kind of split and become mice that survive and mice that die? Or is it that they're actually in the beginning already a little bit different, and you can identify those that are resistant and those who are susceptible before the challenge? And then kind of a very interesting question that stems from this is, if this is the case, where you have resistant and susceptible mice to begin with, would they be the same resistant and susceptible mice in a different challenge? So in the half of mice that would die with LPS, for example, would they be the same half of the mice that would die from another challenge, like starvation, for example? And so we decided actually to start with LPS as a model because it's widely used in inflammation research. It's a very standard model and a lot of standard kind of readouts. And we decided not to start with infectious models because there are a lot of, as a kind of an additional layer of potential randomness in terms of where bugs, pathogens could go in the body, for example. And what we found with LPS was that surprisingly, mice that ended up dying ended up losing less body weight than mice that would survive. It's kind of a surprising finding because we usually associate loss of body weight with poorer health. And, but this is what we saw. And you can see that the mice started out weighing the same. And what we did was at 32 hours, if we separated mice simply by the amount of weight they lost, could we then prospectively predict who would die? And basically what we found was that if we drew a cutoff of about 13.5% and then sealed the envelope in our predictions, we could very, very highly accurately predict who would end up living or dying. So this is now, between 24 and 32 hours after LPS that we were able to make these kind of predictions. So in the spirit of the reverse engineering idea, we were then trying to understand with weight loss, how generalizable that this could be. So it turns out that if you use lower doses of LPS where you kill only about 10% of the mice, or you use higher doses of LPS where you kill 70% of the mice, in every case across all doses, the mice that would end up dying would be the ones that lost less weight. And we could generalize this to females as well. And we could also generalize this to highly divergent genetic backgrounds like BALB/c, so that the mice that die are always the ones that lose less weight. Okay, so here we are. We've kind of identified a parameter that we could measure, and then the question was, well, you can lose two kinds of weight: lean mass or fat mass. Which one is being lost? And so here we used then magnetic resonance imaging to assess the amount of fat and lean mass loss. And what you can see here is that if you look at the rate of fat loss, the decrease in weight loss in the mice that died was driven exclusively by the amount of fat that they lost. So that mice that eventually died, and here you can see that we can resolve to now 12 hours before, 12 hours after challenge, mice that lost less fat mass would uniformly be the ones that ended up dying. And this was not true in the lean mass. So then actually in the course of doing these experiments, we made this very surprising observation that if you MRI'd the animals, these are just, so keep in mind, these are all black 6J animals that we got, that we breed in-house, that we got from Jackson, that are all kept in the same cage, in the same rack, totally normal, a normal chow. That if you looked a couple of days before you gave them LPS, already you could see that there was a difference in the mice that would eventually die in terms of the amount of body fat that they had. This is just across the colony, right? And you could not see this by body weight alone. And so what we ended up concluding was that this, hypothesizing was that this small change in body mass, fat mass in particular, after you hit them with LPS ended up getting amplified. And so that we could highly observe this. And then the question became whether or not the fatter mice just had a different disease than their slightly thinner counterparts. So again, remember that these are all normal mice, right? They're not on high-fat diet or anything. These are just within the normal range of body composition. And so we asked the question, is this in fact two different diseases? Because we have two slightly different groups of mice. So we checked a lot of things. I'm going to show you just some of them, but we checked, for example, whether or not LPS, after you gave it to them, was cleared at the same rate between survivors and those that died. We asked in screening lots of endocrine cytokines whether or not kind of the response after LPS in terms of the inflammatory magnitude was the same, and we didn't find any difference in those either. We ended up actually, because mice that die from LPS die from the initial loss in blood pressure from the inflammatory response, we ended up doing experiments where we implanted sensors in the carotid artery where we could continuously measure blood pressure, heart rate, and other physiologic parameters. And you can see that after LPS, the ones that survived and ones that died had the same kind of initial shock response. And it really wasn't until in the end when they were moribund that we actually saw a precipitous loss in blood pressure. So we don't think that there was at least a measurable difference in all the ways that we measured the inflammatory response that was different in the early part of the LPS challenge. So because the phenotype was composition of fat, then we looked at all of the types of hormones that could control fat in terms of loss of fat or making more fat, so lipolysis or lipogenesis. And so we did this screen basically looking at all of these different hormones. And what we found was that as early as 2 hours after LPS, you could start to see that the mice that died ended up making a lot more insulin in their circulation, and also one of the products of insulin metabolism, C-peptide, was also different between the mice. And so it led us to think that perhaps the slightly fatter mice were more insulin resistant after we gave them LPS. And so in this experiment, what we did was Michael and Kavita did MRI on the animals and then made a prediction based on their percent body fat, who was going to live, who was going to die, and then did a glucose tolerance test to see how sensitive they were to insulin. We also did insulin tolerance tests. And the bottom line is that these mice appeared to be more insulin resistant after LPS. And if you then measured insulin, now this is by ELISA, and at the same time measured glucose, you could see that the mice that ended up dying, like we saw in our screen, ended up making significantly more insulin over the course of LPS, but the level of glucose, surprisingly, was not different, consistent with the idea that they are more insulin resistant. We had previously shown when I was a postdoc in Ruslan's lab that ketogenesis was a very important aspect of surviving LPS challenge. And so we looked at a whole bunch of things. And here I'm just showing beta hydroxybutyrate, which is one of the ketone bodies. And consistent with our findings from many years ago, we did find that there was a modest but significant difference in the amount of ketone bodies that we could detect in the blood. Again, consistent with the fact that mice were less able to engage in lipolysis and fatty acid oxidation. And to kind of, in a brute force approach, test whether or not we could genetically recapitulate these findings, kind of in extreme examples, we made mice that we could inducibly and in a tissue-specific way, in adipocytes, force more fat loss by making them insensitive to the suppression of lipolysis by insulin, and also make them unable to release their fat mass. And you can see that consistent with our overall idea that the amount of fat is really important in surviving or dying from LPS, we get predictable results here, where more fat loss leads to better survival and no fat loss leads to worse survival. And so we think that this little bit of increase in the normal dynamic range of fat composition in normal wild type mice makes them just ever so slightly less insulin sensitive, leading to kind of an increase in insulin that then prevents lipolysis that leads to death. And so we think that this is probably what's going on, that in fact you have two slightly different groups of mice that end up giving you two very different kinds of diseases with the same kind of challenge. So then we asked the kind of an obvious question was, well, can we make one mouse and one disease trajectory into the other? And so, and vice versa, could we push these kind of endotype of mouse into the protected surviving endotype? And so first question we had to answer was, because here usually we group two or more litters of mice together, was whether or not there were kind of cage effects, and if it was all or primarily mice from one cage that was dying compared to another. And so we made sure that that the survivors and the non-survivors was not an inter-cage phenomenon, but an intra-cage phenomenon, so between littermates. And consistent with this, the dynamic range of body fat composition was also not an inter-litter phenomenon, but an intra-litter phenomenon, so that within each cage and each litter, there was a distribution of body fat percentage, which is not surprising, but something that was important to make sure of. And then we wanted to know how stable the percent body fat was as the animal developed. And so what Michael and Kavita did was they MRI'd animals serially, starting from 4 weeks of age. And they've actually done this quite far out now. And what they found was basically that if they looked at the same mouse at 4 weeks of age and plotted against its fat composition at 5 weeks of age, there really wasn't a nice kind of fixing of body fat composition. But what's very striking, as you can see, is that after 5 weeks of age, a slightly fatter mouse stays slightly fatter at 6 weeks, at 7 weeks, at 8 weeks, and this goes, I think we followed this out now to about 6 months, so that something fixes the slightly fatter or slightly less fat composition sometime between 4 and 5 weeks. So the first experiment we did was, can we make a mouse whose kind of body fat trajectory is fixed? Can we change it so that we can alter its sensitivity to dying from LPS? And so what Michael found was a diet, and this is actually it's a high carb diet, that he was able to get mice to be pretty much a little bit fatter than mice on a chow diet. And this, as you can see, is within kind of the 10%-ish normal dynamic range of just normal B6 mice. And this doesn't, and in a way that doesn't affect their body fat. So this isn't like a high fat diet or Western diet where the mice get really, really fat. The body weight's the same, but they have just slightly more body fat composition. And as you can see, the slightly fatter mice then are significantly more susceptible. The other example I'm going to show you, we did several other versions of this, but the other example I'm going to show you is a before birth example where we put mom on a high carb diet. And then he noticed that the offspring of moms on a high carb diet actually had slightly lower body fat composition. Again, this is all within kind of a normal dynamic range without affecting overall body weight. And as you can see, again, it's the slightly fatter mice that end up succumbing to challenge from LPS. And so, we then kind of asked, well, what exactly is then causing this divergence of mice into slightly fatter, not as fat groups? And so we did lots of experiments to try to figure out what would be causing this kind of dynamic range. And it was not food and water intake, activity, litter size, the mom. We were very careful at looking at a lot of different types of things that might end up leading to this divergence in body fat composition. And in the end, what we found was that transit time, so the amount of time that food stays in the gut, was one characteristic of the host that seemed to be correlated with the percent body fat, so that mice that kept food in their GI tract for longer ended up being slightly fatter, which makes sense in calorimetry studies looking at the amount of extraction of nutrients from the stool. This bore out also. Interestingly, the length of the small intestine where a lot of nutrient absorption occurs, you can see it's quite variable, did not correlate well with how fat a mouse was. So we think that the dynamic variability in transit time is probably what's causing slight differences in body fat composition. And so to see if we could manipulate this to get mice to be slightly fatter or not, we ended up using a drug that we use in people to stop the motility of the gut. This is loperamide. And so what we did was in this window where body fat was being fixed, we treated some mice with an agent that slowed down gut transit time, and then washed it out for a week, and then looked to see what we got. And so you can see that the net effect of this is no change in the body weight, but that if we slow down transit time using a drug, we could now make mice slightly fatter. And we did verify with transit time assays that the drug did what we thought it did. And then in these mice that are slightly fatter, they were significantly more susceptible to LPS. And so now at this point, we were thinking, we have this observation that differences in transit time could lead to differences in body fat composition, and which seemed to lead to a different kind of disease after the animal got LPS. The microbiome, which links all of these things, was an obvious candidate. And so we teamed up with my colleague, Noah Palm, and one of his MD PhD students, Mitian, to ask this question of whether or not there was a role for microbiota as the generator of these of this dynamic variability in these traits. And so we did 16S sequencing of survivors and non-survivors, and keep in mind that these are mice that are littermates in the same cage. And what we saw here was that we could actually, that there were differences even between littermates in terms of the abundance of certain types of microbiota. And the top hit was one of the SFB bacteria, Candidatus. And, we verified this by qPCR, quantitative qPCR, that those that died did in fact have a slightly higher burden of SFB than those that survived, and were also slightly fatter. And so the obvious experiment then was, could it be that kind of the random acquisition of SFB that could occur, slightly more, slightly less, be the source of this stochasticity. And the results actually were fairly disappointing. So in these experiments, we used mock communities with and without SFB. And we did not find differences in transit time, and we didn't find differences in body fat. However, the animals that were colonized with SFB were significantly more sensitive to LPS. And this is consistent with other literature in terms of kind of the pathogenic role of SFB in making inflammation worse. But this essentially led us to the conclusion, it would have been much nicer if random acquisition of SFB was in fact driving all these phenotypes, but that there probably was kind of a third group of animals where if they had slightly higher carriage of SFB, they would also be more susceptible. This kind of link between microbiota and transit time, we could not in the end experimentally validate. Okay, so I think what I've showed you so far is we think that variability in transit time leads to variability in body fat, and slightly fatter mice then are more susceptible to LPS. And so then we ask the question, is this host feature that confers susceptibility to this challenge the same? Would these mice also be the ones that would be more susceptible to a variety of other standard challenges that we use in the lab? So surprisingly, with poly-IC, the answer is yes. So this is poly-IC, as you know, is a viral mimetic of inflammation as opposed to LPS, which is a bacterial mimetic. And I'm not going to go through all the data on this slide, but briefly, the same kind of higher insulin with the same glucose, the same manipulations with diet, the same genetic manipulations preventing fat loss or increasing fat loss, all led to the same observation that the mice that would die would be the ones that were slightly fatter. And we were surprised by this finding because of what we had found in 16 when we were doing feeding studies on animals that were either had LPS or poly-IC. But I think here the big difference is that in the previous studies, we were manipulating diet after animals had been challenged, whereas in these studies, there's no such manipulation, right? We're manipulating them before they've been challenged and asking the question, what's the host characteristic that makes them more or less susceptible? Interestingly, with cold challenge, we got the opposite result. So it's not super surprising, but keep in mind, again, these are all mice that are wild type mice on chow diets within normal dynamic range. Mice that were slightly fatter ended up being ones that survived really well when you challenged them with the cold, with no difference in body weight. But if you did, for example, the kind of gold standard model for bacterial sepsis, which is a model where you surgically open the mouse, take out the cecum, tie off the end, poke it, and then let the bacteria spill into the gut and cause sepsis this way. It's not the slightly fatter mice that end up being the ones that die. Neither is this the case when we do another version of shock, which I mentioned earlier in the talk, using an allergic challenge. And neither is it the case when we do acute alcohol poisoning. So there's some other feature here in these challenges that if one existed in them, that would be conferring susceptibility. I do like to point out this cold experiment because the results are just so striking. But if you remember in the LPS setting, when we manipulated fat so that it wasn't restrained by insulin in terms of being released, we got the exact flip opposite result in terms of mortality, consistent with the previous finding that slightly fatter mice would be the ones that live. So how we're thinking about these sets of observations in trying to understand why you can get such different phenotypes with highly homogeneous groups of mice in a highly homogeneous environment is that there is some host characteristic, and we would propose that there may be a host characteristic for many of the models that we use in preclinical testing that interacts with whatever challenge that you're giving the animal to model human disease. And it's this interaction between some host trait and the challenge actually that's the disease. And so the drugs that work really are only the drugs that work for this particular interaction, right? The drug only works for the half of the mice that die. And that this might be one reason why a lot of drugs that look very promising in preclinical models end up not working out very well in human settings. And also that that the question that we might be able to ask more broadly, kind of zooming out of laboratory settings, which are highly artificial, would be that, is it that if we do systematic analyses of these type of perturbations and identify all the different pathways that confer susceptibility across a wide range of environmental challenges, would they all converge on pathways that would be the same in genetically diverse populations like us? Would they be the same variable traits that happen in species that give clonal births, like armadillos? So, these critters are amazing. They give, their clutches are clonally identical, but even within the clonally identical clutches, they're all slightly different in certain ways. And this is beautiful work that is out of Gilles' group that's posted in bioRxiv. And would the pathways that matter all kind of converge in terms of understanding the types of pathways that are required to maintain variability to survive in an unpredictable environment? So which traits would they be? And then interestingly, would there be mechanisms that would generate or maintain this variability? And I would argue that kind of the susceptibility pathways are really not very intuitive, right? So we have one example here of if you're slightly more fat in some kind of challenges, it enhances mortality, but not in other kind of similar physiologic type scenarios, right? Shock. And it's exactly kind of opposite in terms of another environmental condition that an animal might face, the cold. And I think the interesting question would be, are there other differences in parameters that also impose these kind of trade-offs? So conclusion is that the disease model is really kind of a host trait challenge dyad, and it would be important, I think, especially for preclinical models, to understand if any of these traits matter, and that's the actual biology that we're studying. So of course, this is a very similar problem that people think about in various very different fields, like in meteorology. This is the very famous example of where Lorenz, who was trying to model and predict weather, took all the parameters that he thought mattered and then tried to model weather patterns with those parameters. And he found that a very, very, very small change in a weather pattern in one parameter could lead to hugely divergent weather patterns. And we have a very similar example of this, where a very small change in a parameter that matters can lead to highly divergent outcomes. And going back to kind of the title of the talk, this is kind of the same kind of problem that's that people think about in bistable states. So, essentially, what are the characteristics of the tipping points, and would those characteristics be the same for this, the same cat, the same bottle, same box? So with that, I think I have about 10 minutes or so left for questions. This is the group. Very, very happy to work with all these amazing people and our funding. And I'm very, very happy to take questions.